Hi, AP Bio. The topic of today's notes is eukaryotic gene regulation, and this connects to topics 6.5 and 6.6 in the updated AP Biology coursework. So before this video, you should have done this worksheet with me where we introduced gene regulation in organisms. Bozeman has a great introductory video on this, 031 gene regulation. I also recommend watching before this video. And so far we've discussed prokaryotes, which as usual are more simple. So we've talked about the operant. The focus of today's video is going to be on eukaryotes, which is more complex, and there's more places at which we can control whether a gene is on or off, whether or not we're making the product of that gene. And so basically we're talking about epigenetics here. Epi means on top of. So I love this quote, DNA is history, not your destiny, because based on your environment, some of your genes could get turned on or off, and that's going beyond just the code in your genetics. We're going to talk about all those ways today. And so far you should feel pretty comfortable with all of this stuff, operant, promoters, things like that. Some of that's going to hold true with eukaryotes as well. But when we express different genes in different cells, we can actually specialize the entire cell and also control when specific enzymes get created or not to save energy for our body and things like that. So this picture is so big, had to cut it in half, but this is straight from your textbook. And this is representing an entire eukaryotic cell. So imagine this being all one piece. And so eukaryotes control gene expression at every single level, from pre-transcription to post-translation. So we can control the signal. Do we get a signal to transcribe or not? We can modify the chromatin to make it easier or harder to access a certain gene. So if the gene's available for transcription, then our transcription factor is actually binding and allowing RNA polymerase to transcribe that gene. Then we process the RNA that we've talked about extensively. And then when we get to the cytoplasm, how quickly are we breaking down that mRNA? How much are we letting it be translated into protein? Then we might even degrade that protein after. So lots of steps. So first, let's start with DNA. So remember that inside the nucleus, DNA isn't just chilling in chromosomes. It's actually in this bundled up ball of chromatin. And it's double-stranded, of course. And remember, there's hundreds of millions of base pairs in your genome. So a significant number of DNA. And how that all fits, if we were to stretch it out, it'd be like a meter long. So how it all fits is we condense it really tight inside the nucleus. We wrap it around these little proteins called histones that help organize it, kind of how like paper towels come on a roll and how like toilet paper comes on a roll, or like we wrap yarn really carefully. It doesn't come in just like this huge bag, right? And so same thing with DNA, but even more efficiently. And so DNA and these proteins called histones make a chromatin. So whether or not we allow that DNA to be accessed in the first place, it's one way we can control genes in eukaryotes. So most of the DNA is unavailable for transcription, meaning it is wrapped so tight that no RNA polymerase can get in there to transcribe it in the first place. And so we have these things that can make DNA harder or easier to access. So here's the first one. The addition or removal of acetyl groups from the histone proteins that DNA is wound around leads to tighter or looser packing of the DNA. So we call it heterochromatin when it's more tightly packed and it's unavailable for transcription. Means we do not have any acetyl groups attached, but we do have methyl groups. So I typed these on the screen. An acetyl group is basically this addition of this tiny little compound, CH3CO. So if those are not there, and instead we have methyl group, which is a little group of CH3, then the DNA is unavailable for transcription. So if you like doing color coding on your notes, heterochromatin you can do in red, okay? And in particular, methyl. When we methylate DNA, it is unavailable for transcription. It's like turning it off. And then if we remove acetyl groups, okay, acetyl groups usually help out. So if we remove them, then we will be not able to access that DNA. So if I color code this one green, but then I can like draw a red X through it. If I take these acetyl groups out or I add methyl groups, that's going to make really tightly wound DNA that is unable to be transcribed. Euchromatin, on the other hand, by the way, U means like good, and so or new, and so this is like, yay, good DNA that we can transcribe. So this is when it's going to be more loose, it's less tightly packed. RNA polymerase and transcription factors can get in there and transcribe genes. So again, acetyl groups, we're just adding this little CH3 acetyl group in little spots to the tails of DNA. And we're removing methyl groups. And when we remove those methyl groups, then we're able to access it. So just again, pointing out what this looks like. Here's really, really, really tightly wound DNA. Remember these little purple balls are histones. Those are proteins that we wrap the DNA around. And when we acetylate it, they repel each other. And when these acetyl groups repel each other, it's because it's like this little polar group that has a oxygen on it, which we know is very polar. They repel each other, so these little clumps of histones spread out, and then we can get in here and transcribe it. So acetyl on, methyl off. That's how I remember. Acetyl on, methyl off. Acetyl on, methyl off. What's really cool about this is that this is happening constantly throughout your lifetime. You can acetylate and methylate different parts of your DNA. But sometimes it's kind of stuck and it stays on or off depending on the acetylation and methylation. And methyls can stay even through cell division. And so if you have parts of your DNA that are methylated and turned off, when we replicate the DNA and then split it up through mitosis, those methyls go with it. And so that same gene is methylated even in the daughter cells. So once you methylate something, it stays off unless you get some intense signal to remove it. In some cases, it's permanent. Methylation can also be passed on if methylation happens to the DNA in an egg or sperm. So you can actually inherit genes from your parents that are already turned off because of methylation. And this can happen due to the environment during fetal development. And so depending on what a mother does during her pregnancy, her baby, the embryo, which then becomes a fetus during the 9 months of the gestation period, based on the environment that she's in or what she's eating or stress levels and things like that, certain genes can be turned on or off even within the womb. And what's really cool is, say you have identical twins, and they can actually experience a microclimate in the womb. And some of these identical twins, even though they have identical DNA, might have certain DNA not methylated, so that can be turned on, and some DNA methylated, turned off. And that's called either genomic imprinting, so you're like imprinting on the fetus while it's still inside you. And the sperm, again, too, is genomic imprinting. It's methylated when the sperm gets passed on. Or sometimes it's called epigenetic inheritance. And so, again, some of these can get altered, right? So it's not like a mutation. You're born with that methylation, but it could get removed within your lifetime. So anyhow, a little side note, but pretty cool. It's called epigenetics. I also have a little joke about this on the cover of your Unit 6 packet. So hopefully you'll understand that when you look at it now. All right, let's move a little more quickly through some of the rest of these. So other pre-transcriptional controls, meaning we're in control of this before transcription. We can control the transcription factors present in the cell. So remember, transcription factors bind to the DNA upstream from the actual gene we need and help RNA polymerase bind. So we can control whether or not we have those transcription factors there or not. And so this is a major way that we help control. So a lot of cell signaling will ultimately end up telling transcription factors to go bind to DNA at a certain spot so that we can have RNA come and start transcription. And so again, these transcription factors can bind here at enhancers, which are even farther upstream, or they can bind here at the promoter. Again, that's right in front of the gene we actually need. So after transcription, we have post-transcription, but before translation. As we know already, so this is quickly going through it, we keep exons, we get rid of introns, but alternate splicing, we can combine exons in different arrangements, keep some introns. So here is one really nice example of how we have the primary RNA transcript with the exons here. And in this one, we actually cut out exon 4. We don't use it. But in this one, we leave in 4, but cut out 3. So these are going to be closely related proteins that are similar, but not quite the same. And this is for something called troponin. RNA interference. These are microRNAs, so little tiny pieces of RNA that are produced that bond with specific transcripts and block translation. So RNAi, they do exactly what they sound like. They interfere with the translation of mRNA. So these little pieces, they're small, and they would be the complementary code to the mRNA. So if this is the interference RNA here, and this red mRNA transcript is the gene that we're trying to interfere with, the interference would have to have complementary base pairs, okay? It would bind to it, and then it would not be able to get translated, and then it would get broken down. So remember, we have these exonucleases just waiting to chomp apart mRNAs so we can reuse nucleotides, or it just prevents it from binding to a ribosome. So in both cases, it interferes with the translation of that RNA transcript. All right, post-translational control. So this is after translation. Protein targeting, signal sequences on polypeptides. So again, that primary structure, the amino acids, will target the production of the polypeptide into the cytoplasm or into the endomembrane system. So we've talked about this a few times. This is helping control where this final protein is going. If it's made on a free ribosome in the cytoplasm, that protein's going to stay inside the cell. If that protein needs to be exported out of the cell, for example, insulin, because it needs to go through the bloodstream and help bring down your blood sugar, then that would go to the endomembrane system, which remember is the rough endoplasmic reticulum. We call it this because it's connected to the nucleus and the smooth endoplasmic reticulum. So you need to pause the video. It's a good picture reviewing that here. Okay. Lastly, protein activation and degradation. So proteins for certain cellular pathways are made in inactive forms that are only activated when specific signals are received. And so we can have proteins like an enzyme, for example, kind of remain in its dormant stage. So what we could do is we could put like a little block. It's kind of like when we were talking about a methyl group. We can put a little chemical in the active site of an enzyme, so it's ready, it's there, so we can use it as soon as we need it, but it's not doing things when we don't need it to or want it to. And so once a signal is received, again, signal transduction pathways, be a really good idea to review those at this point in the year, can remove kind of that little inhibitor to an enzyme. We learn about enzymes and cell energy. You might remember inhibitors of enzymes. So So we might remove that inhibitor so the enzyme can do its work. Okay, and that's just one example. Proteasomes are organelles that target and destroy proteins that have been tagged to be degraded in the cell. And so if we no longer want a protein because we don't want its function or structure anymore, proteasomes can break it down back into amino acids so they can be used again to build a new protein that we do actually need. So I went through this a little fast because I wanted to save you some time. But basically, we just want to understand that there are many places at which we can control whether or not we actually go from gene to protein, control how much protein is being made and for how long. And then eventually we're going to want to maybe break that protein down. So if you need a little bit more help on these topics, please read 18.2 and 18.3 in your textbook. The pictures are great. And so even just skimming those and reading the captions and all the labels is really helpful. There's a note guide for this in your packet. AP daily videos, there's 3 total, 2 for topic 6.5 and 1 for topic 6.6. These topics pretty much overlap because cell specialization is due to gene expression. So these are kind of like, how do we actually regulate gene expression and how does that lead to cell specialization? There's a few really good Amoeba Sisters videos, gene regulation, order of the operon. It's gonna be a little bit more about prokaryotes, and then epigenetics, getting a little bit more into eukaryotes. I have many more resources besides this that I'll post on the videos page, but these would be a good place to start if you needed more help understanding eukaryotic gene control. And I hope that was helpful.